Live with the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh, it's Science Tonight. Now here's your host, Chris Smith.
Good evening, everybody. Welcome to the show. Welcome to Science Tonight. I am your host, Chris Smith. I work at the North Carolina Museum of Natural Sciences, and it's great to be with you for another Thursday night on Science Tonight. Listen, we've got a great show for you tonight because we've actually got a handful of guest experts who are going to be joining me on the program in just a moment to talk about a new discovery that was pretty much right in our backyard, their backyard, and one that happened in the most, maybe not the most, but in an unexpected way, I think, which I guess some of the best discoveries always happen that way. But thanks for joining us on tonight's show. Remember, everybody, that as we go throughout the program, you can interact with us, whether you're watching on YouTube or on Facebook. Use the chat box, use the comments, say hi, tell us where you're visiting from. If we were at the museum, I would ask you where you're visiting from. We're not doing that right now. Tell us where you're watching from if you happen to be local, because if you're local, you may one day see one of the specimens of tonight's topic. Anyway, so let me know where you're from, or, just like drop a waving hands emoji to say hi. It's always good to hear from you. And most importantly, later in the show, we'll be taking your questions. So use the chat box. Let us know what your thoughts are. We're keeping an eye on that. And later in the show, we will do audience Q&A. So stick around for that. Let me bring on tonight's guests. We've got a group of fun guys who are going to be on the program tonight. And that's also because we have three fungus experts or fungi experts. I don't actually, would you be fungus experts or fungi experts? Let me introduce them. First off, I want you to meet Dr. Van Cotter. Dr. Cotter has been an herbarium associate with the UNC, University of North Carolina Chapel Hill Herbarium since 2014. And he's on call as a fungus expert with Duke University and NC State, a veritable mycological expert. Van, welcome to the show.
Thank you. Hello, everybody. Good evening.
Next up, meet Corbin Bryan. Corbin is a senior at UNC Chapel Hill, studying biology and German, and is from Durham, North Carolina, just up the road. Hey, Corbin, welcome.
Hey, thanks for having me tonight. Nice to see you, and welcome to everyone who's watching.
And then last, certainly not least, is Dan Meyers. Dan is also a senior at UNC Chapel Hill, and where'd my notes go? And he's the Mary McKee Felton intern at the UNC Herbarium since 2018 from Holly Springs. Dan, welcome to the show.
Hey there. Thanks for having me.
I'm really excited to have you all on because the three of you made a pretty interesting and actually pretty important discovery. But on Science Tonight, before we do anything serious or important, I like to warm everybody up. We're going to get the crowd warmed up. We're going to get us warmed up and into the program with just a little bit of trivia. So for tonight's game, I thought, okay, what would three experts on fungus, mushrooms, mycology, what kind of trivia questions could I put together for them? And my first thought was, I'll find some pictures of like mushrooms and we'll just see if they can identify them. That's no fun. Of course, you can probably identify them unless they're just like really rare kinds of things, right? That's not going to work. So then I thought, okay, what's the most famous mushroom in the world? The most famous. And if we can get the trivia pulled up, the most famous mushroom, I think is, next slide, I think it's Toad from the Mario Brothers franchise. So I've prepared 3 questions for you about Toad. You can work together, you can work against one another, however you'd like to play it. but this will be the test. So the first question, you can see it and the answer choices here on the screen, and I encourage everybody who's playing at home to let me know what you think the answer to the questions are. Which Mario franchise game did Toad, pictured here, make his very first appearance? Was it the 1985 Super Mario Brothers, the follow-on Super Mario Brothers 2, or Mario Kart 64, released in 1996? Corbin, what do you think?
Well, I think I'm going to have to go with answer choice B this evening.
Okay, Super Mario Brothers 2, and sounds confident. Dan, what do you think?
I think I'm going to agree with Corbin there. I know he's definitely in 64, but I'm pretty sure he came before that.
Okay, so we're in agreement. Dr. Cotter?
Well, they're going to be on a better foundation about this than I'm pretty certain it's not A, which would leave me B and C. And given my expert colleagues here, I'm going to go with B as well.
All right, our three mushroom experts are all saying answer B, Super Mario Brothers 2. Let's see the correct answer. I'm very sorry. It is, in fact, the very first Mario Brothers game released for NES in 1985. Yeah, in fact, on the next question, I have a screenshot of where you can find Toad. So let's take a look at that. And our next question, there's Toad right there. Toad featured at the end of each level in that original game. And his character is the faithful guard of one of these major players in the Mario franchise. Does he guard Bowser, Princess Peach, or Koopa Troopa? Although I say guard, he never did a good job. He kind of always got caught and had to be rescued. Dan, what do you think?
Answer choice B, Princess Peach.
Confident. Okay. Van, how about you?
Of course, last time we were confident, but I like peaches. I'll go with Princess Peach.
2 votes for Princess Peach. Corbin, do you agree or disagree?
I got to say, I agree with Dan and Van here. I'm going to have to go with B as well.
B, Princess Peach. Let's see the correct answer. Toad is the faithful guard. Yes. Princess Peach, fantastic. You're 1-1. As we head into the third and final question, that means you've got to get this one right in order to walk away as a part of the Science Tonight Hall of Fame. You did get one question correct, so that means we won't be putting you in the Science Tonight Hall of Shame. Congratulations. But here's your third question. Is Toad's mushroom cap, I hope it's actually called a mushroom cap, part of his head, or is it a hat? In the Mario universe, is it part of his head, or is it just a hat? Let's start. Dr. Cotter, you get first go at this one.
Oh, dang. 50-50. I'm going to go with B. It's a hat.
It's a hat. Toad can take it off and on. All right. Corbin, what do you think?
I'm feeling like it might be B as well. I'm going to have to go with B.
All right. 2 votes for B. Dan?
I want to say B, but we can't all have the same answers. So I'll go with it's part of his head.
Gonna go with this part of his head. Any particular reason? Like, is there something about a mushroom cap that a fungus expert would lead towards it being part of his head and connected for a cartoon?
Well,
version?
as far as real fungi go, it should definitely be connected. But video games can be kind of different. So
All right.
Yeah,
Well,
mycologically,
let's see the
I agree
correct
with
answer.
Dan.
Well, there you go, everybody. It's actually both, because video game designers and the animators behind so much of the Mario franchise could never really decide. So there's an entire universe where he can take the cap off and put it back on. By the way, Toad is bald underneath the mushroom cap. And, yep, at other times, actually in an interview, a video game designer with Nintendo said outright that it is not a hat. It is part of his body. So I'm not really sure what we're supposed to do to that. But what that does mean is that you got 2 out of 3 correct. And we're going to put the 3 of you into the Science Tonight Hall of Fame. Congratulations. Very well done. That probably wasn't quite the mushroom quiz you thought you'd get this evening, was it? Everybody's just like laughing, like, oh my gosh, this is ridiculous. I wish he'd ask us about actual fungus rather than.
And my little brother's
Mario
gonna
franchise.
kill me for getting that one wrong, the first one.
Oh no, I didn't mean to get anybody in trouble by being on the show tonight. Well, perhaps I should steer the show onto topics on which the 3 of you are definitely an expert. Corbin, I want to get started with you because when I was hearing about and reading about this new mycological discovery, it seems you were the first eyes to be laying on a new specimen. Can you tell us what a chip cherry is and how you came to know about them?
Sure. So the chip cherry is a fungus, one might more formally call Leratiomyces ceres, but chip cherry works just fine as well. It's a small mushroom with a pretty bright red cap and a kind of a white khaki colored stipe, I would say. And it grows on chips, wood chips and mulches and decaying woods of all sorts. And how I came about it isn't, well, maybe it is remarkable just on how, I guess, serendipitous it was. I was just walking into the herbarium to see Dr. Cotter and Dan and drop off a couple of specimens, and I found it fruiting out of a mulch bed next to some viburnums. And there it was. And I thought, well, you know what? Nothing is quite as fun as having a nice little, look at a fungus, seeing what it is. Always like a little puzzle to solve. So I figured I'd bring it inside and see what Dan and Dr. Cotter could make of it.
Dan, what did you make of it?
Yeah, so at first I saw the cap and the white stipe, and I was like, oh, that's probably a russula. There's a bunch of those. There's kind of a species complex going on for red russulas in the southeastern United States that make them sometimes pretty difficult to figure out which is which. A lot of times you need molecular evidence to really confirm a species identification in that group. But then we flipped it over, looked at the underside. The gills were black, which is definitely not something that russulas would have. They would have a white gills. So we were like, this is something else. This is something interesting. And then just kind of started throwing back and forth some ideas of what we think it might be. And Stropharia came up kind of as something that I thought, but we weren't really certain. So we started going through some online keys and things like that to try to confirm the identification.
And Dr. Cotter, you having, I'm imagining, seen lots of fruiting fungus since becoming an herbarium associate just a few years ago in 2014, being an expert on fungus generally, did you have like an immediate sort of thought, I know what that is, but it's not supposed to be there? Okay, sorry everybody. I think Van, I think you were muted. I couldn't tell if there was audio going out, and I just couldn't hear it, or if that was all of us. Folks watching in the chat, let me know.
Nope, that was my error. I apologize.
Okay, no problem.
I'll keep
it looks
my
like
mic on.
we're good. That's fine. We'll take it from the top again. Corbin brings in a species of mushroom, and we're not really sure what it is. He doesn't recognize it. Dan doesn't recognize it right off. Isn't sure because there's some scientific wiggle room going on in species identifications in a similar group of mushrooms. But then you get your eyes on it. What were you thinking about?
Well, like Dan said, the genus Stropharia came to mind as a possibility, but the bottom line was it was new and different, something I had never seen before, and that's always exciting. And it's a really striking, beautiful fungus. So it's one that if you had come across in the past, you would remember. So we knew right away it was something new and exciting. And we put aside what we were doing, dropped what we were doing, and all hands on deck to figure out what this interesting and beautiful mushroom was.
So, Dr. Cotter, give me a little bit of insight then on how you would go about identifying a mushroom. Because I know being out in the woods, going on nature walks and hikes all the time, or just, seeing them out in a yard or a park, there are many different kinds of mushrooms and like shapes, colors, sizes that you see. But so often I see like really general characteristics that seem similar. Like, oh, all of those, while they may be different species, all have brown on the top. Or I've seen several different kinds of mushrooms that have red on the top. But I guess these are all actually different, and there's ways to differentiate them better than just whatever color is on the top fruiting body there.
Oh, there are many things that come to mind, features that are useful. Color is one of them. Size. Habitat's really important. Is it in a lawn or is it in the woods? And as you get to know mushrooms, you always bounce what you're seeing against what you know from the past and try to fit it in like Dan would with that one. It kind of looks like a red russula. So we started there, but then turned it over and the spores are black, not white. So went down a different path with it. Artificial intelligence is, you got to take that with a grain of salt. A lot of people use like iNaturalist and ask iNaturalist, what is this mushroom? Probably 9 times out of 10, iNaturalist is going to get it wrong. In fact, with the chip cherry, there were 11 reports in iNaturalist in North Carolina, the chip cherry, and none of them really were confidently chip cherry. One was a stinkhorn. I think one was a russula. One is even a bolete. So again, yet to date, artificial intelligence isn't where it needs to be.
So the internet wasn't as great a resource then. Took a little bit of work on that one. So how did you come to decide that this was in fact this chip cherry, Leratiomyces, this not really probably found in North Carolina yet species of mushroom? Dan?
Yeah, so we kind of started doing some of the work. We looked at some of the basic morphological characteristics, matched them up with sort of keys online, while searching with images through iNaturalist or things like Google Photos can be useful for a starting place. Kind of as Van was saying, you've got to really double check them against verified keys and things like that. And those can be either found kind of in books or online. A really great resource for American mushrooms is mushroomexpert.com. So yeah, we started kind of going through that stuff, working our way through some dichotomous keys, and then checking out some of the other features. So there's obviously the cap color, stipe color, spore color, things like that. But you also kind of have some other features down the line. One of which included a potassium hydroxide reaction. That's where you kind of take a little dropper of potassium hydroxide and place some on the cap. And I think we've got a picture of that actually, if that could be shown at this point. It's a really cool reaction. Sometimes there's no reaction to potassium hydroxide on caps, but sometimes like this, in an instant, it turned kind of grayish on a otherwise brilliantly red cap. So that was a really important feature. And then we also looked at it microscopically, so under a microscope, and checked out some of the spore measurements and verified spore color, things like that. And those are as well. And both of those can be really important features when identifying a fungus. And they matched up perfectly with Leratiomyces ceres. So we had a pretty solid identification on that. But then in addition to that, we got some molecular evidence, and Van or Corbin can go more into that for sure. I'm pretty sure Van did the sequencing on this one.
Okay, so is that like just taking a piece of tissue and running it through the DNA sequencer?
Yeah, so thanks to the generosity of Dr. Vilgeli's lab at Duke, I just took a small fragment of the gills from this mushroom, extracted and amplified the DNA, and there's a DNA sequence, the internal transcribed spacer region, that is used as a barcode for fungi. So once I had that sequence, compared it to the database, and it was a spot-on match. There was no doubt about it that it completely confirmed our identification of this fungus as Leratiomyces. So we were quite confident without the DNA, but slam dunk once we had the DNA.
And then 100%, just about, I guess, that you've got something that's new to North Carolina. No other, I guess, real records of this species of mushroom fungus in the state. So, Corbin, maybe you can tell us a little bit about what does that mean for us to have this new species pop up?
Sure.
Like what makes this chip cherry special?
Well, the fact that it hasn't been here, means that it had to have come here from somewhere else, right? And so we're not exactly sure how it got here, number one, but it also points to the fact that this mushroom's distribution is growing. It was previously in areas of Australia and in the northern United States, and now it's making its way down here. And, we're not exactly sure why or where it came from, but that's what this really shows us here, I would say.
Okay, so it's a non-native species of fungus is that to all of North America? Or do we have occurrences? Are they like pot marked? Does it seem to always travel on loads of mulch or something like that, and that's how it got to UNC in the first place? I don't know who would be the best one to take that one on.
Well, it's
I can
pretty
go ahead and
clear.
do it.
Okay, go ahead, Van.
It's pretty clear that it's native to Australia, so that's where it started out. Described in 1888. It took it 75 years to get to California and about the same amount of time to get to England. And it took it 70 more years to get to New York, 2012, first seen there. And then Corbin's discovery here in North Carolina in 2021. So it's definitely a globetrotter. And we tried to track down how it showed up in Chapel Hill, but Dan and I went out to the mulch facility that UNC has, And it was clear all of the mulch is generated locally from local trees and whatnot. So it's a mystery. We really don't know how it ended up here in Chapel Hill.
What do we know about how a fungus like this moves around in the first place to even show up in Chapel Hill if it's not been seen like places between us and New York? I think you were saying.
Right.
So yeah. Oh,
Go ahead, Dan.
I can take this one, I guess. Yeah. So fungi have a number of ways of moving. They both grow vegetatively through their substrates, and that's in the form of mycelium. And that's a little bit harder for us to track. And if we don't really see fruiting bodies, it's difficult for us to track that. And we're not really sequencing random piles of dirt or piles of mulch. So we don't have good records of the vegetative spread of fungi often. However, we also get movement of fungi through spores. And those can travel through the air and sometimes can travel pretty far distances through the air. They're very small. That picture that we pulled up a little bit earlier was at 400 times magnification. So they're a few micrometers in length and width. So those can get carried along on wind pretty easily. There's also definitely some movement of mulch around in the United States. But yeah, what was really puzzling for us was kind of how it was in the Chapel Hill area, especially if we were getting all of our mulch sourced locally. So that was kind of, Van and I did a little bit more investigation into that, but with not a ton of success. But that also kind of shows that fungi sometimes aren't noticed as much. And I mean, we're grateful to Corbin for keeping his eyes open as he was entering into Coker Hall that day, because Van and I had taken that same path inside but completely missed it. So that was a very, very just cool discovery, and also just kind of shows how fungi can be there even for people who know what to look for and usually are actively looking for and can go unnoticed. So that also is definitely how they can move around such great distances without a lot of people noticing their occurrence as they move.
Well, okay, so you bring up a great point. Two fungus experts blew straight past it, headed into the building. Corbin, though, what made you stop and notice it in the first place?
Well, I think if I remember correctly, I think I was having issues getting into the building because I think the doors were locked or something because of the new regulations. And so I think I had to move from one door to the other, which forced me to walk past the mulch bed, which it was growing in. And otherwise, I think I might have missed it as well. You're not really, I don't think one expects mushrooms to be growing in mid-December. I certainly didn't. I wasn't out looking or hunting or anything of that sort. So I think it was just because the doors were locked.
Yeah. You couldn't get in, so you had to look around for a minute, try to find the right door, and then there's this new thing. And so, I'm always fascinated by how scientific discoveries are made. So often they come from rigorous experimentation. I think of physics experiments at CERN or Fermilab, right? It's experiment after experiment over years and years, and then they come to some new conclusion about the universe. In this case, though, you essentially tripped over this thing, and it could have been so easily missed. I mean, is there a sense of that when you sort of realize what you've done, that you've actually picked up this new species and what that could mean for the world of fungus in North Carolina?
Well, for me personally, when I just thinking back on it, I'm kind of surprised I picked it up because typically, what grows in mulch isn't always the most compelling, fungal find. So I'm definitely kind of, I still think there's a little bit of a wow factor there. I thought it was like a Sathrella or something, and those are a dime a dozen. So I'm pleased, in short. Yeah, it was pretty exciting.
Dan, what about you?
Yeah, I think, I mean, first of all, it's just a great find. And I mean, I think as far as knowing what you have as soon as you pick it up, there's definitely sometimes when you can do that, but other times you definitely can't. So it's kind of a little bit of a journey, and as Corbin was saying, a little bit of a puzzle to figure out what fungi are what and what you've picked up or are looking at. And yeah, sometimes you need to take it back to the lab and do a little bit more investigation, and that's always fun as well. But yeah, I mean, and it's important for sure because you get, especially in the herbarium, you get vouchered specimens. So you have things that can be set up and left for scientists in the future to look at, as well as you get a kind of like permanent record of a species in a time and a place. And that can be very important for ecological studies in the future, for tracking pathogens or non-native species as they're moving around. So it's just a cool process, as well as it's very important. And I mean, probably up until 50 years ago or so, a lot of people thought herbariums were kind of old dusty libraries of dried up plants and fungi. But now with the advent of kind of DNA and molecular biology, they're really quite a treasure trove. And there's a lot of data that you can still get from these specimens, which is always great.
Yeah, when I think about the herbarium, of course, there's a direct connection to what we do at the Museum of Natural Sciences as a natural history museum with an extensive research collection. Actually, I don't think we have very many plants or fungus. I think our herbarium at the museum is quite small compared to what y'all have got going over at UNC. Dr. Cotter, do very many mushroom discoveries happen this way?
Oh, definitely. To Corbin and Dan's credit, they're very good observers, and that's what it takes: good observation and paying attention to the world around you. One thing that's really exciting is, like, in teaching a lab, like in mushrooms, say for a course of students at UNC filling in, and then to learn a week later, seeing them on campus, and all of a sudden they're noticing all these things, all these fungi out there that they used to walk by. So just making people aware of their natural environment is exciting to see them then follow up and express gratitude and having their eyes open to see this natural world that's part all around us, but often ignored. So yeah, discoveries of fungi are made like this, absolutely. And I wanted to make one other point. This fungus would probably still just be in Australia if it weren't for the human impact on the planet. So some fungi really take advantage of human activities, and this is one of them. So we humans have changed the planet in a way that has allowed this fungus to move around the world.
Okay, yeah. I mean, you make a good point. It's cool to have this new occurrence of this, you said globetrotting species of fungus. Is it important that it's here, though?
Is it important? I think it's important to know that it's here. And of course, invasive species are a concern of all of us, be it invasive fungi like the pathogen for Dutch elm disease or chestnut blight. So a fungus that moves out of its natural range into a new range is always a concern. In this particular case, it's a decomposer. It particularly seems to flourish in human habitats and not so much out in the wild. Obviously, it was there and can be there, but it really flourishes in human-derived habitats, especially wood chips. So this particular fungus moving around probably isn't of the level of concern that having an invasive plant pathogen would be.
Okay, well, that's good to know then. We don't have to be too worried about it. But I guess, folks, it's something for people to keep an eye on, at least if they're hanging out around UNC Chapel Hill. More of these mushrooms, I guess, should be popping up, right? Could people expect to see these start showing up, around Chapel Hill? Would we expect it to spread? Do we know anything about it that could indicate where it's going next?
I find it likely that.
Go ahead, Dan.
Sorry about that. Yeah, I find it likely that it'll be seen around Chapel Hill again, and I think there are definitely some people out there looking for it, especially with all the publicity that it's gotten, which is pretty cool. I would also expect that it's probably in the North Carolina Piedmont and will start to be seen in other places. And I mean, part of that is definitely sort of like an avenue where citizen science can help track the movement of this fungus a little bit. I mean, again, don't necessarily, especially if you're using an app like iNaturalist, just immediately agree with the first suggestion. But there are definitely some ways that citizen science can help us track the movement of non-native species, as well as native species, and just getting some of their distributions and occurrences. As for future, it seems like it does well spreading, and I would expect it to continue probably.
There you go, everybody. Keep an eye out. That'll be what we'll say. Just keep an eye out. And even if you don't see Leratiomyces, this new one, you never know what you might see anyway, right? There's probably all kinds of great new mushrooms out there waiting to be discovered. There's a great question here in the chat, and looking at the clock and the number of questions that are coming in, I think I'm going to start with Cindy's question, and we'll roll over into audience Q&A right now, because Cindy's asking, does the triangle area have a high diversity of fungi species? And I'm curious, do we see new ones like this popping up with some frequency?
I'll take that one. Yeah, we do have a pretty good diversity of fungi, not just in the Piedmont, but if you take North Carolina as a whole, for sure, from the coastal to the mountains. So we have a varied series of habitats in the state, and the fungal diversity reflects that. Sometimes new fungi are identified that went under old names, like John Gibbs, a 13-year-old who has actually brought some specimens of mushrooms into the museum and added to your collection, found a Neolecta, which is a little yellow fungus. And it turned out it was a new record for North Carolina. It had always gone under a different name, but again, doing the work, working it up, including the DNA, it turns out that John, who's only 13, excellent observation skills, has added to the mycoflora of North Carolina.
Oh, you gotta love that story. That's incredible. Just good observing, just paying attention, right? And then knowing
Absolutely.
to, I guess, send an email to you, Dr. Cotter. So the next one that I've got here, Bev is asking, what's involved in registering your discovery so that others around the world know? I'm going to
Well,
guess
there,
that the three of you wrote a paper.
well, we haven't
Yeah.
reported it in this. Go ahead, Dan.
So we've reported it in the sense that we've accessioned it into the herbarium at UNC, as well as I think there might have been an iNaturalist observation of it. For citizen science, iNaturalist is probably one of the top ways to go. That's a great way to record your findings. If you want to actually get into the process of making specimens out of your findings and getting some higher levels of identification and ID confirmations, usually, especially, I mean, around the triangle, there are herbaria and there are experts that you can kind of get in contact with, and especially if you're interested and want to make specimens and want to donate your own specimens or keep your own herbarium, that's an option for anybody. So yeah. As far as the paper goes, yeah, we didn't write a paper about this new state record. That's not usually kind of in the range of what I guess we would publish a paper on. But this specimen now exists in the herbarium and can be loaned out or requested by researchers all over the world. So.
Very
And
interesting.
if I could put a plug out there for citizen scientists, do more than just take a picture of the top of the mushroom. Like even the Mario character, if you just have a top-down view of the top of his hat or head, it's not going to tell you everything you need to know about it. So if you're interested in contributing, you really need to take pictures of all the salient features, the top, what is like underneath, side view, different stages, note the habitat. So it takes more information than just a simple picture of the top of the mushroom to figure out what a mushroom is.
Very good tips. Yeah, I've taken mushroom pictures, and they were all of just the bright, colorful top and no other identifying characteristics. I don't even think I put anything to scale on the photo. So that's not a good way to go. So let's see. Corbin, this one might be a good one for you. Could you determine what type of tree mulch the chip cherry was on? Because Penelope writes they understand some fungi are associated with particular trees.
Well, I don't know. Unfortunately, I wasn't able to make it for the touring of the facility, so I'm not sure exactly what kind of a tree mulch it was growing in, but it was certainly locally sourced. So I think a lot of I'm not really sure what the most amount of what mulch is typically made from in the area. So I'll have to turn that one over to Dr. Cotter or Dan, unfortunately.
Dan, do you know after visiting the facility what the species? I imagine the mulch pile was just a mix of a lot of different things.
Yes, it was a mix, but Leratiomyces ceres is also known to grow on conifer mulch, pretty particularly. So at least some part of the blend was conifer, probably. I think it was pine. Some of the UNC grounds people came by and actually went through the bed and started picking out wood chips and identifying sort of the genus of tree that it probably came from. So pine was the confirmed substrate, which was pretty impressive. And yeah, that's definitely outside of my wheelhouse to be able to identify the tree from the mulch, though I'm probably getting a little bit better at it. I took a plant class recently. Anyways, but yeah, it definitely was that. And that's a great question. I forgot the name of the person who asked it, but that's kind of, there's a two-part answer. Yes, so decomposers sometimes will have different things that they favor, different tree species, but then also fungi often associate with plant roots. So there are a number of species that are ectomycorrhizal or endomycorrhizal. The ectomycorrhizal species still produce the fleshy fruiting bodies that we would see in cult mushrooms, and that's why, especially with a number of groups of fungi, when you're trying to get identifications, noticing the local ecology and the tree and plant species in the area that you're collecting can be really important for identification.
Penelope also brought up a good point here in a follow-up comment and wrote, mulch beds can generate heat as they decompose. So could that make a difference as to what grows on them?
I think it
Dr.
certainly
Cotter, do you know
could.
about this? Or Corbin, sure.
And I think it certainly could. And also, as you know, as a fungus, fungal mycelium grows, that's also going to generate heat. I mean, it's typically not enough to, start a fire or anything like that, but I think it could certainly be substantial.
I mean, if something like that were the case, it's interesting to think that it's this mulch bed and the conditions become just right for the chip cherry to fruit in this mulch that's been prepared and put there. And then you walk by it trying to find an unlocked door to get into the herbarium where you're already going to talk about mushrooms. And then there's one that you don't quite recognize. Like there's so many dominoes that fell just right in order to get this specimen in the herbarium and identified, and now as a state record. Like that astounds me that you get everything lined up just right to make this happen. And how many times did it not line up just right before? How many chip cherries were growing in the mulch a year ago versus this year? Who knows? We don't know. But it's cool that we have the scientific endeavor, and that you all had the tools and the techniques available and the expertise to not just sort of toss it in an herbarium drawer or jar, right? I think that's fascinating. Okay, let me get you another question here. Troy wants to know if there are regional species lists that are different between places like Florida and North Carolina. Dan, you're nodding. You want to take that one?
Yeah, so there's definitely different fungal kind of microbiota of different areas. As far as a good list for all of those, I'm not sure how many great species distribution lists there are that are very comprehensive for fungi. That also, but definitely in different areas, especially with vastly different conditions, there will be very different fungal species growing. And definitely part of that is, or part of the reason there aren't great lists is because probably like fungi, sort of as we had talked about earlier, aren't noticed as much as other species, and also they have short fruiting time periods, and usually all we're getting to really see is the mushroom, which is just kind of the sexual stage of the fungus. And that makes it sort of difficult to keep track of a lot of these distributions, but we definitely have seen some regional distributions. Like for example, there's some mushrooms that will grow here on the East Coast that natively don't grow out on the West Coast, and the reverse is true as well. But that's a good question, as well as kind of leads me to a point that I sort of want to make. There's a lot of fungal diversity out there that we don't have recorded yet. So overall, there's probably around 120,000 species of fungi that have been described. And that's a very small fraction of the fungal diversity that's out there and estimated. So there are some estimates that put the amount of fungal species in the world around 5 million. I think that might be a little high, but it's definitely up there. And as far as good estimations go, there are probably more fungal species out there than plant species. And yet there's nearly 4 times as many plant species described as fungal species currently. So it's definitely an area where people need to be out there doing the work, and just looking around in your backyard, or walking by a mulch bed next to the biology building, you might find something new to the area or new to science. So keep your eyes open,
Careful, you're going to get a run on the mulch beds at UNC. People looking for new species of mushroom to ID and get to be on the radio about, right? That's pretty exciting. Dr. Cotter, there's a question for you. Muriel writes that they would like to hear how you amplified the DNA of the mushroom so that you could analyze it.
Okay, well, Muriel, very fortunate to have access to Dr. Vilgely's mycology lab at Duke. So the basic process was to take, again, like I said, just a small part of the gills of the mushroom, put it in an extraction solution where the DNA is extracted, and then to use primers for the ITS region, ITS 1F and ITS 4, that basically flank the area of the DNA one's interested in. And then that goes into a machine that amplifies that particular segment and makes a lot more of it. And then one sends it off. In this case, we sent it off to a commercial lab that actually did the sequencing itself. And then comes back with this long list of letters, the nucleic acids, and you compare them in the database to the ITS sequences that have been filed away and see where it matches.
And then I guess if you get a match, you know what you've got. You could also not get a match, I'm thinking now, and realize that you've got something that hasn't been sequenced yet or not described. And Dan, you were just talking about how there's not been a lot of attention paid to fungi. How robust are these genomic data sets or genetic data sets for fungi? Might be one for you, Dr. Cotter.
I can talk about it a little bit. There's definitely some stuff out there, but yeah, there definitely is not, even for all of the species that are described so far, sequences either of just the ITS portion or of other portions. And the ITS region is really good, especially on a larger group of, like looking at it from a larger group, but say sometimes you probably couldn't tell between individual species with that coding region, but you could definitely tell a genus or a family. But yeah, I mean, that kind of gets into phylogenetics, which you can make good sort of predictions based on the data you have, but having more data is always better. And yeah, so Van did the ITS sequence, and that was definitely enough. And I snatched the label that he had from the herbarium, and you can kind of see at the bottom there all those A, T, C's, and G's, and that's the sequence that Van got there.
Oh, how about that?
So yeah.
Oh, thanks for bringing that. That's cool to see. I kind of expected that it was just like an email from the DNA sequencing company, but there you've got it like in hand to go. I'm assuming it gets folded up, placed in the drawer with the specimen, or filed away with the specimen. Like, yep, that's it. See, I've learned something working at a natural history museum. All this stuff goes. Okay, let's see. Troy wants to know, what is the frequency of reproduction of fungi in general? I don't know if there's millions of species in general might be a little bit tough.
Well, I'll take a stab at that. The frequency of reproduction. One thing about fungi, they often reproduce asexually, and that can be extraordinarily rapid. Like when you think of a lot of plant diseases, they will form asexual conidia really by the trillions, and that's why they can explode and cause really major epidemics on agricultural crops. The sexual reproduction is more modest. For most fungi, not all, it would be like an annual occurrence. Like take morels. A lot of people like to look for morels and enjoy them in the springtime as a culinary delight. And they typically, in their reproduction by sexual spores, it's a one-time-a-year event. It would be every spring.
Okay. All right. There you go. Thanks. Okay. Let's see. Adair wants to know, why are fungi specimens kept in herbariums? Aren't they more closely related to animals than plants? That's a good question. And everybody on the panel just started laughing. So we might have an argument on our hands. I don't know. Corbin, what are your thoughts on this one?
Well, so that is the case. They are more closely related to animals, but that wasn't known until relatively recently in the history of mycology. It's in the mid 20th century. Before that, it was all treated as botany. The study of plants, fungi. It was all under this broad category of botany. And I think for instance, if you look at the Duke University biology building, it has little plaques that are carved in stone, and the one that says botany has a plant and a mushroom next to it. So it wasn't that long ago, but that's the reason.
Should we be transferring all of your mycology specimens over to us at the Natural History Museum? Is that what you're suggesting, Adair? I'm not sure. I don't know where we would put them. Next to the polar bear, I guess.
I
Dan
think,
or Van, you want to add to that? Yeah, go ahead.
Yeah, I was just going to agree with Corbin. It's a historical legacy because fungi and plants have cell walls. That was one of the major reasons they were lumped together historically. And the question was absolutely right. When you look at the tree of life, we're more closely related to mushrooms than oak trees are. So it's a bit of an oddity, a historical oddity, that they're in herbaria. But herbaria, like the UNC Herbarium, is really devoted to keeping those collections in good shape as well as the plant collections. So there's no need to jettison them. They have a good home.
I couldn't agree more with that. All right, Cindy wants to know, and it looks like this will be the last question for the night. Dan and Corbin, you are graduating very soon. Congratulations on that achievement. And graduating with the distinction of having identified a new species of fungus to North Carolina. Nobody else at UNC is graduating with that. So great job. Congratulations. Do you know what you're planning to do next? How about you, Dan?
Yeah, so as far as I know, I'm probably working in an algae lab here at UNC over the summer. But then, yeah, still figuring next steps out a little bit.
An algae lab. Okay. We'll have to have you back on the show and talk about algae soon. Corbin, what about you?
Well, I'm hoping to get a research assistant position. I got a couple of potential ones lined up. Hopefully one working with fungi, we'll see. But that's my plan for the foreseeable future.
Best of luck. Best of luck. And I guess, Dr. Cotter, you'll have a new crop of fungus excited students coming into the herbarium soon then.
Absolutely. Hopefully so.
Well, Dan, Van, Corbin, thank you so much for being on the show tonight, for sharing with us the story of this discovery, a new fungus species for North Carolina, a new state record, for giving us some insight into the world of fungi. Although you said fungi, didn't you? Have I been saying it wrong this whole time? Is it actually fungi?
Your call. Either way is fine.
Okay. Just checking. Thanks to the three of you. Thanks for being here on the program. Everybody, thanks for tuning in to Science Tonight this evening. Hope that you enjoyed getting to learn something new, being curious with me about tonight's topic. Next week, next Thursday night, of course, we'll be back here right again on the museum's YouTube channel. Next Thursday night, I'll be talking with an HIV vaccine researcher at Duke University, Dr. Christine Daniels. So we're going to be talking about using vaccines to end HIV. Don't miss it. We're going to have a great conversation. I hope you'll be here for it. Make sure that you're following the Museum of Natural Sciences on social media. We're at Natural Sciences on Facebook, Twitter, and Instagram. And of course, you can subscribe to the museum's YouTube channel right here. That way, you always know when we're bringing you great, new, fun, and exciting science content. That is what we do here. So everybody, big round of applause for our three guest speakers. I'll say thanks again one more time for sharing all of your knowledge and expertise with us. And everybody, take care, stay safe. We'll see you again real soon. Good night.